You're listening to the Effective Statistician Podcast, a weekly podcast with Alexander Schacht and Benjamin Pieske, designed to help you reach your potential, lead great science, and serve patients without becoming overwhelmed by work. Today I'm talking with Miguel Pereira about Bayesian approaches in early clinical research. So stay tuned for a really nice episode. And now some music. Miguel has done a lot of research in this area and has also a lot of applied knowledge. So he's the perfect person to talk about this topic. And we are not just talking about this topic in this podcast episode, but he will also be a presenter at the Effective Statistician Conference that happens on April 25th of 2023. This 5-hour conference will feature lots of speakers beyond Miguel. We'll have about 10 presenters that speak for about 15 to 20 minutes plus Q&A. And so you will have a lot of opportunity to interact with the different presenters and ask your questions. And it will be quite a nice thing, not so kind of boring with lots of long presentations, but more kind of shorter presentations so people can stay engaged. And especially for these virtual ones, it will be a lively thing. So stay tuned and register for this conference. I'm producing this podcast in association with PSI, a community dedicated to leading, promoting the use of statistics within the healthcare industry for the benefit of patients. Join PSI today to further develop your statistical capabilities with access to the video-on-demand content library, free registration to all PSI webinars, and much more. Head over to PSI website at psiweb.org to learn more about PSI activities and become a PSI member today. Welcome to another episode of The Effective Statistician. Today I have Miguel with me as an expert in Bayesian statistics for early clinical research, and we'll speak specifically about oncology today. That's definitely an area where I have only a little bit of insight, so I'll be learning a lot today as well. Hi Miguel, how are you doing?
Hi, how are you? Doing well, thank you.
Very good. So maybe for those who don't know you, maybe you can introduce yourself first.
Yes, so my name is Miguel. I'm based in Cambridge, UK, and at the moment I work as a statistical consultant for a company called Cogitars, which is based in Germany, and we provide consulting services mostly in the design of Bayesian clinical trials in early clinical development. We don't just do Bayesian statistics, but that's the main focus of the company.
Yeah, and we actually had Oliver on the podcast already at least once, probably more than 100 episodes ago. So if you go back, we talked about working for big companies, for small companies, for CRO, and being a freelancer. And now he's growing his own business, Cogitars, which is great to work with. It's a great consulting organization, especially in that area, and that can help quite a lot.
And the focus is not just on providing services, which would be the main focus of CROs. Our focus is not to be a CRO, but to provide expertise in novel statistical methodology and in developing novel methods for novel designs, because drugs are different and have different requirements, and that's our main focus.
Yeah, and I know that you also do quite a lot of actually CRO oversight work for...
Yes, that is correct. Yeah. When we work with clients, we partner almost as their statisticians, in-house statisticians, as we were their in-house statisticians, and we work with CROs and try to articulate the interaction between the client, what we're doing, and the CRO.
Yeah. Okay, let's talk about early phase development, and specifically we'll focus today on oncology. What are the typical questions that come along?
So in early phase development, and this is something that goes pretty much across the board, regardless of whether we're talking about oncology or any other area, it's mostly about safety. We're talking about first in human trials usually. So phase 1, we want to know we want to find what is the highest level in general of a drug that is still considered safe in humans. So part of it is how can we define the maximum acceptable dose, or MTD, maximum tolerated dose, while keeping the patient safe. There is one thing which is particular of oncology, and which I would say specifically of oncology, which is usually the first in human trials are in people that have cancer and not in healthy subjects as in other areas, where a phase 1 trial would be in healthy individuals, because we're effectively not that interested in efficacy yet, but we are interested in safety. And an interesting thing that comes from that is that we are not only treating people that are ill. Because we are treating people that are ill, we also want to reduce the amount of subjects that are exposed to subtherapeutic levels of a drug. Because if we're talking about a drug that could change the landscape of that disease, we want to make sure that these people are also benefiting from it, because usually they are late stage patients that don't have many choices in terms of treatment because they've basically exhausted all the lines of treatment that they had.
Yeah. And it's also, especially when you think about oncology, lots of these indications are rare diseases. And that's another reason why you usually want to restrict your number of patients, because it can take forever to recruit lots of patients. So when we think about this and we want to understand, okay, what is maximum tolerated dose? How is that traditionally?
So the most traditional design, and this is a design that is specific to oncology, is the 3+3 design, and it's extremely easy. And a lot of people listening to this will recognize what it is, but it can be explained in 30 seconds. So in a trial, you enroll patients at the first dose level, you enroll 3 patients, record the number of dose-limiting toxicities or DLTs. Usually grade 3 is what we're aiming for. And we see how many DLTs occurred in a cohort of 3. If 0 DLTs occur, we can escalate to the next dose level. If 1 DLT occurs, we should enroll and expand on that, and we will have 3 more patients at that dose level, meaning we have 6, and we check whether have DLTs or not. And if 2 or 3 DLTs occur, we stop at that dose level. And this is a very simple design. And it's so simple that I've just explained it to you, and you don't have to be a statistician to understand it. It is anyone can understand and implement it.
You stay at the same dose. Imagine you have 3 patients, one of these has a DLT, and then you enroll 3 further on the same dose, and then none of these have a DLT. Then you would still go to the next dose?
Yeah, so then if you have 1 out, which would mean if you enroll these 3, you would have 1 out of 6, because you recorded 1 in the first cohort and none in the second 3-patient cohort. And so 1 out of 6 would be you can escalate to the next dose level. And effectively, the cohort sizes is essentially 3 or 6. Okay. That's why it's called the 3+3, because 3 is the baseline or the normal cohort size, and then eventually you might expand to 3 more, which would be the 6th, so 3+3.
Okay, and if you have 2 or 3 in the first step set, get it, then you directly stop there and see you have reached your one step
MTD.
Beyond the maximum.
Exactly. So you would say, this is the MTD. We cannot go more, so we cannot continue the dose escalation. And this is the maximum dose that we are going to give these patients in, say, phase 2 trial, or eventually in another expansion cohort that is outside of this 3+3 design.
And you mentioned grade 3 toxicities. For those people that are not working in oncology you don't have the typical kind of severe and serious adverse events. You have toxicities and all the different.
No, you also consider severe and treatment associated adverse events, but you have grade toxicities that goes from 1 to 4, and usually it effectively has to do with either the duration or the severity of a certain symptom. It could be a headache. So if you have a mild headache for 3 hours after you've taken the drug, that would most likely be considered a grade 1 adverse event. But then if you have, say, an immune reaction to a compound, and for some reason you had almost cardiac arrest, or you had to stay in hospital for 24 hours under observation, and a special treatment has to be administered, you most likely go into the realm of the grade 3 or grade 4. However, it's not very hard to get into a grade 3, because if it's something that already, in some cases, if it's something that already interferes with your daily life in a significant manner, in some cases that could be considered a grade 3. So there is, I don't know off the top of my head what are the full criteria for a grade 3, but they are fairly well defined in terms of intensity and severity.
Yeah. How long is the follow-up time usually to check for these toxicities?
So that depends essentially on the drug being administered. That decision is usually made by the medical team or by the clinicians that say, for this specific drug, given the regimen or the number of cycles that are being administered, this being in a chemotherapy G-related setting, that depends on more on the clinical decision rather than a standard follow-up period. So we could have a follow-up period of, say, 3 days, but we could also have a follow-up period of, say, 21 days or 7 days after the last administration. So, for example, let's say we have a cycle of 3 administrations of a drug, which they're administered once every week. So this could be a period of 21 to 28 days, according to what the follow-up is, where we give the first administration and we record if there are any DLTs. Second administration, we record if there are any DLTs. Third administration, we record if there are any DLTs. So this would be a 21-day regimen, for example.
Okay. And when you then check for these DLTs, so basically you need to basically have a database log after each of these steps where you get all the information in, do the analysis, check for these things, and then you have.
You don't necessarily need a database. You don't need necessarily the database log, because usually you do the database log when you're doing more the final analyses or an interim analysis. So in this case, it depends on how the assessment of DLTs is done and the dose escalation meetings occur. Okay. If, for example, we have a certain drug where a patient comes in, is given a certain drug, the patient is assessed for 7 days after, or the cohort is assessed for 7 days, we could potentially have a dose escalation meeting at the end of those 7 days, and we would decide whether we escalate or do not escalate. That is something specific of these dose escalation trials, where you need to get the data, analyze the data, and make a decision, and then decide whether you do escalate or don't escalate. And in a way, that's why the 3+3 design, which has many limitations, but it's very useful, because if you just know the clinicians have been observing the patients, so they know the number of DLTs and they just know whether they can escalate or not, the amount of statistical work is minuscule.
You just need to make sure that you have all your data in place.
Exactly.
Yeah. Okay, very good. Let's talk a little bit about, you just said it's pretty simple and easy, but what are the limitations of it?
Yes, everything tends to be a trade-off. First, you trade simplicity for, this case, less accuracy, less flexibility, and effectively, with simplicity as well comes the limitation of not being able to do things like you are stuck with the dose levels you have. So if you had to find 5 dose levels at the beginning of your trial, those are the dose levels you're going to assess. If for some reason you think, perhaps the last dose level or the second to last dose level might be a bit too much, you cannot just go and say, we're just going to reduce that dose. You don't have that flexibility. Also, your cohort sizes are 3+3. You cannot do 4, you cannot do 2, you cannot do 7. And that's usually a thing that clinicians have a hard time understanding, because the reason why it's 3+3 is because we're targeting a certain proportion of DLTs that come from the fact that it's 3, because 1 out of 3 is a third, and 1 out of 6 is about 16.7%. And these DLT rates are the ones that are usually accepted in oncology. So our targeting rate would be about 30 to 33% of acceptable grade 3 DLT rate for a cancer drug. So the 3+3 is completely fixed. Clinicians have a really hard time understanding that. And on top of that, we've talked about inflexibility, being stuck with the same dose levels. We also have the fact that it's less accurate. In general, the targeting rate of a 3+3 design, meaning if you do a simulation, the proportion of trials or the percentage of trials that get it right, to get the MTD right, is 35 to 40%. So this is just a little bit more than a third of trials get it right.
So if we talk about accuracy here, what exactly is accuracy here?
So in this case, accuracy in the targeting rate is how many times in a trial, or when you're simulating trials, how many times do you get the right MTD?
Okay, if you assume of these 5 MTDs that you set, and you say, okay, you basically know, okay, this one is the right one out of these 5. In about a third, you get exactly that right one, and in the other cases you would get a higher or a lower one.
Correct. Yeah. Okay. And for example, and I was doing some calculations, and if, for example, the real DLT rate is 0.5, so if at those levels you have a 50% chance of having a grade 3 DLT, which is very high and way above what you would accept, you have an 11% chance of escalating.
Okay.
So you should not only stop because it's already too high. It could, in 11% of the cases, so if you simulate 100 trials, in 11 trials, you will escalate. So you are, and effectively would be, presumably the next dose would be considered to be toxic, and you would stop there, but you would be declaring an MTD that is too toxic. On the other hand, you can be unlucky and you could go to the first dose level, everything's fine, second dose level, and you observe 2 DLTs just because you were unlucky. And you're done. And the drug does not get you, and you cannot test other dose levels. So in a way, it can go to effectively, it's bad both ways, because you can be taking dose level that is too toxic for a phase 2 trial, or you might not even do a phase 2 trial. You might just kill the drug because you were able to escalate to a level where you can see some efficacy.
Okay, very good. That makes a lot of sense. And I think as statisticians, you can really easily see these kind of limitations. I would say one of the biggest limitations is that it's really difficult to pick in between, because if you think about it, probably there's some kind of curve, and you probably want to get close to it as possible, isn't it?
And in fact, that brings me to another limitation of the 3+3, which is it's algorithmic, meaning you have those rules and there's no flexibility around that. And also, when you look at those level, you are just looking at that information. You don't care about what happened before at all. So, for example, let's say you reach the 3rd dose level, and in the first dose you didn't see anything, the second dose you didn't see anything, and now you see 2 DLTs. It's very different from a trial where the first dose level, you 1 DLT, you expanded, etc., but you were able to escalate. The second dose level, you see another DLT, you expanded, but you managed to escalate, and now you see 2 DLTs. If you look at the history behind it, those are 2 very different scenarios, which the 3+3 design completely ignores.
Yeah, that makes a lot of sense. When you have at the next dose level, the first 2 DLTs, you can more likely assume that maybe you could go further. Whereas if you have, as you said, in the second scenario described already, 2 DLTs as 2 different lower dose levels, then it's just from a maximum likelihood perspective, I would say it's much more likely that you have reached your MTD.
Exactly.
Yeah, very good. That makes a lot of sense. So let's bring in Bayes now. Bayes seems to be one of the really nice things that help us to be much more flexible and bring in some modeling and these kind of things. How does that work with using Bayesian approaches here?
I guess first thing is, and that's one of the things I like about Bayesian, I find it, and this is not specific to clinical trials, just Bayesian statistics as a framework or as a school in statistics, I find it a lot more intuitive, not just to me as someone who does statistics, but also to what I work with clinicians. And I didn't say that in the beginning. Originally, I studied medicine, so I have a medical background. And that's the way I think. For me, thinking sometimes about p-value, it's very convoluted when you're thinking about its definition. Whereas with Bayesian, and I can ask sometimes questions to people that know a lot about statistics, and they don't know the definition of a proper definition of a p-value, because when you think about it, it's not intuitive. And with Bayesian in general, I find that things are a lot more intuitive, and it's more in line with the way our brains work, in the sense that we, as people in our decision making about whether we go shopping right now because it's sunny, so it's not going to rain, we just assess probabilities, and not in a formal way, but informally we are assessing probabilities. And that's what Bayesian formalizes in a mathematical way, is probabilities with the use of probability distributions. And that's why for me, then moving to doing work in statistics in general, and now in clinical trials, using Bayesian statistics is what I feel is natural. And on top of that, because we're talking about probabilistic decision-making, we know that's the biggest advantage of statistics. We can include prior information.
Yeah, I think that is a real nice thing. Yeah, that basically exactly. You build on the information as you collect it. So as you just mentioned, you learn something on the first step, you learn something on the second step, you maybe go into the experiment with other knowledge about similar compounds or from maybe some tox studies or whatsoever. And you could leverage that. Yeah.
And effectively, because we are working in a phase 1, mostly in a phase 1 setting or phase 1-2 settings, our sample sizes are very small. And in order for us to have what we would call enough statistical power, we need to leverage prior information in order to be able to make good inference. And with this, I'm not implying that we cheat, because a lot of people say you can use prior information so you can influence your final result by using a heavy prior that will just overshadow the data. We definitely don't do that, and we have a lot of ways of working around that to make sure that the prior is not influencing the data in a biased manner?
Yeah, well, it's a little bit of a philosophical question. Yeah. If you put a lot of emphasis on your prior, you need to have a lot of belief that it's correct, or a lot of evidence that it's correct. And if you're borderline, it looks like we are pretty close. We just need a little bit more evidence. Yeah. Then, of course, your additional study doesn't need to be super big. Whereas you start with nothing and you don't have no clue of what's really going to happen, then of course your study needs to be much bigger because you get more.
In the use of prior information, doesn't have to be very informative. And the example I like to give when people come with this argument is, okay, if I ask you what is to estimate the prevalence of a disease, let's say, what is the prevalence of, let's think of something like during COVID times, what was the prevalence? Let's forget about incidence, prevalence at any given period of COVID, which at some point was between 10 and 20%. The use of prior information can just be, I know that it's less than 50. And I'm not saying it's between 10 and 20, just I know for a fact, or I'm very sure, I don't know for a fact, but I'm very sure that it's less than 50. So this is a way of incorporating prior information, which everyone would find, I'd say, reasonable, which would increase our statistical power and our ability to make inference without biasing the data.
Yeah, completely agree. That's a good example. Okay, very good. Now, if we apply these new Bayesian approaches, what kind of flexibility do they give us?
Yes. So I think there are multiple ways of applying Bayesian statistics to trials, but given that we talked about the 3+3, and what we usually use in the case of dose escalation trial is a model-based approach, or a Bayesian model-based approach, where effectively we do a logistic regression model where we are modeling the probability of a DLT. Mm-hmm. And that probability of a DLT is a function of an intercept and a dose. And effectively, so if you think alpha plus beta times dose, our beta parameter, that's what we want to estimate as being the DLT rate associated, or our beta parameter is what we call the toxicity associated to a certain dose. And that is what we are estimating in our model. And in terms of flexibility things, one is this being a model-based approach, and this being a regression model, we can make predictions as to what will happen in the future. So I observed the first 3 dose levels, and this is one of the advantages is we're taking into account the entire history, the entire data that's been observed. We can estimate the probability of DLT at the next dose level without having observed it. And of course, we have the way we're doing this is we have a probabilistic decision rule where we say, specifically for cancer trials, our targeting rate, or we want a DLT rate which is less than 33%. So ideally a DLT rate which is between 16.7 and 33.3%. And the way we decide is if the posterior distribution of the DLT rate, so if you look at distribution, the probability that the DLT rate is greater than 33% is greater than 25%, we stop. So effectively, and I know I'll repeat this because this statement is complicated to understand in itself. Essentially, I look at the density of my distribution of the DLT rate. If the density is set at 33, between 33 and 1, so the right-hand side of my distribution, if that is more than 25%.
Yeah, if in this densities, also more than 25% of the density is beyond this.
Exactly. Is above 33%, then we stop at that dose level. Or we stop at that dose level because that's already considered too toxic, or we don't escalate because that's what we obtain when we run our model for the next dose level.
Okay.
So that's how we make our decision. And of course, this is what is usually done in the oncology setting. But for example, in other settings, we've used different ways of doing this decision rule, which could be 50%, and obviously will not be 33, it could be 20, or it could be 50, depends on the setting, the drug, and what we're modeling. But this is the way we apply Bayesian statistics to a Bayesian statistic or a logistic regression model to the dose escalation setting. And going back to what you mentioned in terms of flexibility, even though we can have preset dose levels, because we're doing a model that considers a continuous scale for the dose, we can effectively test any dose. So if I say that if I've observed 10, 20, and 30 grams of a drug, and I make my predictions to 40, and it's telling me that 40, I can escalate from 30, but 40 is too much.
You can also do all the predictions for everything between 30 and 40.
Exactly. So I could say, let me try 35. Oh, 35. My model is telling me that I can escalate to 35. And so I would try 35. And that's where a lot of the flexibility comes from, because effectively in our trial, when we're designing the trial, we don't really need to declare all the dose levels we're going to test. We can say these are the ones we're going to test in principle, but we can define intermediate dose levels according to a certain set of criteria. Or you can just say that our increments will never exceed a certain amount. And this is the reason is because if we are making predictions in a linear regression model,
you want to have a
predict
little
too
bit
far
of a
ahead in
safety
time.
margin.
As we know, if you observe between 10 and 30, you're not going to make accurate predictions for 60 because it just doesn't make sense. But if we have that, with how much we can escalate, then we can make predictions and define what would be an optimal, in theory, define what would be an optimal dose level.
Yeah, that makes a lot of sense. And you could also go back to an intermediate, isn't it? So if you, let's say, if you go to from 30 to 38, and then you see, oh, that is too toxic, you could also go back to something like 35. Could you?
Exactly. These models allow us to de-escalate, which the 3+3 doesn't. So the 3+3, it's a one-direction model. And with the BLRM, we're not just escalating, but we can de-escalate. And of course, like the 3+3, we can stop at a certain dose level and do an expansion cohort to gather more information, and then make a decision whether we want to try those levels that are higher or lower. So in that sense, there is complete flexibility. And effectively, we're just including all the information in our trial. Also with the advantage, because we are using a Bayesian setting, we are not doing any hypothesis tests. So it's continual assessment of what is going on.
Yeah, it's much more of a estimation topic rather than a testing topic.
Correct, exactly.
Which really is the origin of the problem. Yeah, you don't want to test the MTD, you want to estimate the STD. If you also mentioned you can be more flexible with a number of patients. So you could have, instead of 3, you could have, I guess 1 is probably not a good solution, but a 2 or 4 or 5.
Yeah, so effectively, we can use any number of patients in the model because we've got a prior and we're just adding information to that prior. Technically speaking, in Bayesian, you were doing an interim analysis every time you observe a patient because you're just adding more information to your model. Of course, that's not ideal. We don't want to do that. But you could say, for example, that the first 2 or 3 dose levels, because they are really low, you enroll less patients, say 1 to 3 patients, but then in the higher dose levels, you start enrolling more patients, so say 3 to 6. And you don't need it doesn't have to be 3 or 6, it can be 3, 4, 5, 6. It doesn't matter because effectively you are just collecting more and more information and just enriching the amount of information you have about that drug at different dose levels.
Yeah, where you get faster to the higher levels where you want to get to. So you jump over the initial kind of lower levels that you just set for safety faster, and especially with these probably more rare diseases, that is really important.
Even not being rare diseases, even common cancers, say colon cancer or lung cancer, which are very common, by using less patients in the first cohorts, you are reducing the number of patients that are being helped.
For sure, that's the other thing. Ethics is for sure another topic. Just was going from the, let's say, more operational point of view. Okay, very good. Now, you mentioned at the beginning with these 3+3 designs, the beauty is its simplicity. Now, what you just described obviously is everything but simple. At least it's simple as you explained it, but I'm pretty sure if you implement it, that doesn't look simple.
Yes, I would say it's 3+3. It's as simple as we've talked about, and there's not much more that people haven't heard about it that we haven't mentioned here. It's really easy to understand and know pretty much everything there is to know about it. But with Bayesian, of course, you need more training. You need to know a little bit more about Bayesian statistics and how it's implemented. And I have 2/3 challenges with Bayesian statistics, which I don't think it's about understanding because, as I said, I think it's very intuitive. But it pertains to, the programming itself. Essentially, we are talking about using somehow more specific or more niche types of programming, which involve using either JAGs or STAN and previously WinBugs. So the model coding has got a specific syntax, which adds to what you need to know. And also on top of the coding, because we are using, in order to assess the operating characteristics, and we need to test some scenarios. And the reason why we need to test data scenarios is to make sure that the prior is not influencing the data in a way that is biased, for example, or that doesn't allow us to escalate. The prior itself wouldn't allow us to escalate to the last dose level. So we need to run these. And this can be very heavy in terms of programming, because effectively, the more flexible you make it, the harder it is to simulate a trial under these rules. And what we do with the operating characteristics, we simulate trials, usually 500, and that's how we define how many trials we are getting the targeting rate right, are defining an overdose or an underdose, or trials stop early, for example. And all of this is very heavy from a programmatic standpoint.
Yeah, so being the studies, the protocol itself is already quite a big of programming.
Yes. Yeah. And I guess you could say that either at that stage or at the stage of the SAP. But for us, it's mostly at the protocol stage. We put all the operating characteristics and the data scenarios in the protocol so that it's clear that with what we did and that the prior we have works well. And this brings us to what I think is the main challenge in using Bayesian statistics. It's the prior, because we effectively need to translate clinical information, and we have either need to look at the literature or have information from the clinicians and then transform that into prior distributions. And that is naturally something that I personally find it's the hardest thing to do in Bayesian, and perhaps the biggest hurdle in terms of using Bayesian statistics, because statisticians can very easily get the programming in understanding Bayesian statistics. That's very easy. But then getting information from an article or from a clinician, which says, oh, I think the DLT rate at 20 micrograms will be 40%. And that DLT rate at 50 micrograms is going to be 60%. And then you need to get that information and say, I think my prior probabilities or my prior distribution for the DLT rate is this. And that is what I think is hard and the greatest challenge when you're starting with Bayesian.
Yeah, the prior elicitation topic
is correct.
Definitely a big one. Very good. By the way, Miguel will also present at the Effective Statistician Academy that is coming up. So if you haven't heard about this academy yet, then I would say, hey, head over to the academy, the conference yet, then head over to the Effective Statistician landing page, our homepage, and check out and register for this. The conference will take place on April 25th of 2023. As we are recording this, we are at the end of 2022, and I'm really excited about this. And then you will not only hear what Miguel has to say about Bayesian statistics, but you will also see a lot of these kind of different things. And you probably get, with a couple of these things that we talked about, posterior and then prior distribution, and the probabilities that something is bigger than whatsoever, it's sometimes easier to see. Head over to theeffectivestatistician.com and check out the Effective Statistician Conference. It's great to have Miguel presenting there.
Thank you for having me. It's good fun.
Yeah, it will be fun. It will be a pretty nice event. We have so many speakers, great speakers lined up. Can't wait to get to it. Okay, very good. In terms of Bayesian statistics, is there any key things that you would see as a listener to take away from the call today?
One thing is in the 3+3, starting with the 3+3, it's still used very widely in oncology, and it's also known to be a very suboptimal methodology. And even though people might not want to invest the time to learn the BLRM or model-based approaches, there are other approaches that can be used, like the MTPI2 or the Boin, VOIN, which is that one is effectively Bayesian 2, that can be used as a transition to methodologies which are more optimal in terms of defining the MTD. But in terms of Bayesian, what I think is useful to know is that there is a lot of value in using Bayesian methodology in trials in general. And effectively, one very well-known trial used the Bayesian methodology, and that is the Pfizer BioNTech COVID-19 vaccine study. First publication they did, and this was a phase 1, 2, 3 trial. That was a Bayesian methodology, not a Bayesian logistic regression, but a different method. And the trend seems to be that Bayesian methods are going to be used more often, because there are a lot of advantages in terms of operating characteristics and flexibility and ease of use, and also by being intuitive. So I think I don't think that there is a battle between frequentist statisticians and Bayesian statisticians. I don't see myself as a Bayesian statistician. I do Bayesian, or I do a frequentist method when it's appropriate.
Completely agree. It's about using the right tool at the right time and not be dogmatic about it.
Exactly, because I think there's a lot of value in using Bayesian in early stage development because you have small sample sizes. But then as you get to phase 3, where the sample sizes are much larger and you want to have more control of the operating characteristics, where a prior doesn't have that much influence, probably using frequentist methods are probably a better option for many different reasons. It's more about using the most appropriate approach rather than saying, I just do Bayesian or I just do frequentist methods.
Yeah. And so that's a great way to actually end the episode today. Enrich your tool set. If you're working in these areas, get to know about all these kind of different things and tune in for the Effective Statistician Conference on April 25th. Thanks so much, Miguel. Thanks for being on the show today.
Thank you very much. This was amazing. I don't get tired of speaking about Bayesian.
Don't forget to register for the upcoming conference, the Effective Statistician Conference, where Miguel and many others will present about their research and how you can be a really, really good statistician. I actually renewed my membership very, very early in 2023 already. This show was created in association with PSI. Thanks to Rain and her team at VVS who helped with the show in the background, and thank you for listening. Reach your potential, lead great science, and serve patients. Just be an effective statistician.